ended up in Firenze with Ivano Bettini, because he was an expert on metalloproteins. And I was fortunate enough to obtain a grant from the German Cancer Aid. And I went to Ivano for 2 years for the following reason. In Bernhard Kepler's group in Heidelberg, we had been working on these two complexes. These were ruthenium complexes with very high anti-tumor activity in vivo. You can see they are structurally very related, the only difference being the ligand. This is an indisol ligand here, and this is an imidazole ligand. When you dissolve them, they form these nice brown solutions. They have these abbreviations KP1019, KP480. And in Heidelberg, I had a hunch because there was a dramatic difference in their toxicity, despite more or less identical anti-tumor activity. KP1019 showed minimal systemic toxicity, while the toxicity for this compound was extremely high. And I had a hunch that it had to do something with their interaction after intravenous interfusion. And this is basically what happened when you took these solutions and mixed them with human serum or albumin or transferrin. The solution would turn green, which is a nice thing for chemists to observe. The main difference, however, was the time which it needed for that color change to occur. For the very non-toxic complex, it only took 5 minutes for that color change to occur, while for this toxic compound, the green color would occur after around 5 to 6 hours. So with this hypothesis, I went to Ivano Bettini's laboratory. What you see down here is an incubation study of KP1019, the indisol compound with human serum. This is a crude separation on LPLC at that time. And because you have this nice green color, you can follow it at 585 nanometers, you see it's binding to transferrin and albumin, basically in a ratio of 10 to 1, which is the ratio at which it occurs in human serum. We also then, with a collaboration with Edward Baker in New Zealand, showed that this complex could bind to proteins such as transferrin. There are multiple binding sites. This complex then always binds to histone on the protein surface. And I could show when you then acidified this protein ruthenium complex, ruthenium complexes would come off again. So this was a reversible phenomenon. Strangely enough, later Bernhard Kepler managed to find a reformulation for KP1019, because by then the patent was quite old, and it was taken to the clinic with the help of a biotech company in the US, Niki Pharma. It's now renamed NKP1339, and a phase 1 is about to finish in this year. So that's how basically my interest in albumin started. And when I approached and went back to Germany to join the Tumor Biology Center, which was set up in 1994, I wasn't really coming from the approach of tumor targeting. I was thinking more of reducing toxicity. Following the proverb primum non nocere, first do no harm, which is, by the way, an ethical standard of the Hippocratic Oath. Albumin, as you know, is the most abundant protein in the bloodstream. 40 grams per liter, comprises 55% of all serum proteins. So we have around 200 grams circulating in our blood circulation, and there's another 200 grams at least in the extracellular tissues. So all in all, depending on body weight, you will have 400 to 500 grams of albumin in our body. If you isolate it, it's a nice crystalline powder. Commercially now, basically it's available as a 20% solution. It is indeed a drug, a blockbuster drug, and used for, these are just some of the indications where it's used for, as a blood substitute for severe burns, and we use it to treat tumor patients who had a severe weight loss and compensate their albumin loss when they have cachexia. This is the X-ray structure of albumin. It came out in public domain at least around 1997. It's a nice heart-shaped molecule, has 3 domains, and it is per se the transport protein in our body. It transports fatty acids. These are the 5 binding sites shown here for myristic acid. It binds essential metal ions such as zinc or copper. And of course, as you know, in these 2 sites here, the Sudloff binding sites 1 and 2, multiple drugs bind here in a physical manner. And bilirubin, the breakdown product of heme, binds in this region very tightly because it's a very toxic compound. So you see a natural detoxifying function of albumin. It is also, once it's taken up by the cell, an abundant source for covering the amino acid needs of a cell. Now, 1994, albumin was not mainstream research for drug delivery, but there was some data, although limited, that albumin was accumulating in tumors of animal models. Hans-Jörg Sinn in the German Cancer Center in Heidelberg was one of the pioneers. He labeled albumin with indium, injected that in rat models which had different tumors growing in their hind legs. And in this sarcoma model, 23% of the injected dose after 72 hours was found in this tumor. This, by the way, is the liver here, and this is the heart. So even after 72 hours, a lot of albumin is still circulating in the bloodstream. This is in our own work done with collaboration with Dr. Kai Licher in Berlin. We used a different technique, near-infrared imaging, labeled albumin with a cyanine dye, and then injected that into a tumor-bearing xenograft mouse. This is the xenograft. It's a colon cancer. As comparison, this is the uptake after 24 hours of the free dye. And you can see there is no tumor selectivity. If it's bound to albumin, you have a nice tumor imaging. The uptake of albumin is not restricted to mice or rats. It also is present in humans. This is a recent publication in 2006. What you're seeing are the albumin levels in 260 tumor patients. The norm level is 4 grams per liter, and as you can see here, the major amount of tumor patients have depleted albumin levels, which shows that they are basically taking albumin out of the bloodstream to cover probably the need for amino acids. From that, basically, 2 strategies evolved of how you could use albumin as a drug carrier in oncology. The first is to covalently attach a drug or a peptide to albumin. You can enhance this by increasing the amount of drug bound, and in addition attaching ligands, which, for example, would target receptors or antigenes on tumor cells. The other strategy uses a physical attachment of a drug to albumin. I'll talk about this development first because it was the first one which reached the market. This is a technology called Nab-technology, where albumin binds and then drugs bind to albumin and then form nanoparticles. And as an extension of that technology, recent research has focused on these kinds of strategies, antibody fragments, either through a small antibody fragment or a peptide are bound to albumin. So that would be, for instance, a single-chain antibody. And you substitute the IgG portion through albumin. This is the development of Abraxane from the inventor Neil Desai. It's a very simple technology. You take Taxol and albumin. Taxol, of course, is totally water insoluble, but when you mix that and pass that through a jet under high pressure, these albumin Taxol nanoparticles are formed, and that is what is in the vial. It's often seen as a breakthrough for nanotechnology. But one has to be aware the mode of action is not via a nanoparticle. Once this is injected into the bloodstream, it disperses rapidly into albumin Taxol particles. And this is what reaches the tumor, not the nanoparticle. That's important to keep in mind. An advantage, of course, is that this formulation avoids the use of chromophore, which you have to use when you apply Taxol. It's interesting to look at the preclinical data of Abraxane. This was the name it had in preclinical development, Nab-technology. And what you're seeing are results of a xenograft experiment in a mouse model, a direct comparison between paclitaxel and Abraxane. Now, the MTDs between, this is even the older name, ABI-007, are actually not that high. It's about a factor of 2. So you're administering Abraxane at twice the dose of paclitaxel. And this is a dramatic difference you see. There's basically, this is the control, the tumors are growing. Paclitaxel is basically inactive in this model, but the Abraxane formulation at twice the dose has resulted in complete remissions. When you look at how much more Taxol is taken up by the tumor, it's shown in this graph. This is over time, and these are the concentrations of paclitaxel, and you can see this is the area under the curve of paclitaxel, and this is for the Abraxane formulation. And you can see the difference actually at this equivalent dose is not very high. You only have a 1.3-fold increase in the amount of paclitaxel actually reaching the tumor on an equivalent dose. So if you multiply that by that factor, it's around 2.6 or 3. That seems quite surprising because getting 3-fold more of a drug into a tumor, you would not expect this dramatic difference in tumor response. But this has been repeated several times in different models. This has to be taken as true. Consequently, it was taken into the clinic. This is the registration study, a comparison now in humans, 260 milligrams per square meter of Abraxane versus the classical dose for the chromophore Taxol formulation. This ratio is actually lower than in rodents. And what you're basically seeing here is the response rate for all the different patients. There were about 200 in each arm for their different, they were first line, second line, anthracycline exposed, and so on. And you're seeing basically the response rates shown here in this gray color are always higher for Abraxane. And this then led to the approval at the end of 2005 for Abraxane for metastasized breast cancer. In 2012, In combination with carboplatin, it got approval for first-line therapy in lung cancer, non-small lung cancer, and also more or less at the same time for pancreatic cancer together with gemcitabine. This was based on a more or less 2 months benefit in overall survival compared to the single drug, carboplatin or gemcitabine. And so as you can see, it's doing quite well commercially. And if you look at the pipeline, Abraxane is now belongs to Celgene. All these compounds here, for instance, docetaxel, rapamycin, they are all lipophilic compounds. This technology can only be used for lipophilic compounds. That's one of the major differences to the technology which I'm going to present now. And that's the covalent attachment of a drug to albumin, which we focused on beginning in 1994. This strategy actually emerged later, 1998, but it is now our core technology. We develop prodrugs which have a protein binding group, a linker with a cleavable point, and a drug. It's injected directly into the bloodstream, binds in situ very quickly to albumin. So the low molecular weight prodrug becomes a macromolecular prodrug. This accumulates in the tumor, and then there are 2 ways the drug can be released, either by an acid-promoted mechanism or through enzymes which are overexpressed in the tumor. If you look at the amino acid sequence of human serum albumin, there's nothing special. There are 585 amino acids. What is special for a circulating protein is this one. It's the cysteine 34 position, because this is a free tile group. You can see that in the model here of the X-ray structure. That is the cysteine 34. And that accounts for 90% of the tile content in blood. If you measure the tile content of human serum, it's around 400 to 500 micromolar. 90% of that is due to this cysteine 34 position. And by the way, this cysteine 34 is the most reactive cysteine in the bloodstream. So the consequence was to use a tile-binding group which, when injected into the bloodstream, would rapidly bind to this position. So we developed, over the course of time, and this is now the final product, this one, abbreviated doxo-EMCH. It's the 6-malamidocaporyl hydrazone derivative of doxorubicin. This is the hydrazone bond. This is doxorubicin. This is the spacer, and the malamide group. This is a typical group which reacts rapidly in a Michael addition with tile groups. We have meanwhile shown that it shows significant superiority compared to the clinical standard doxorubicin in 8 mouse tumor models. They're listed here. I'll show you 1 or 2 in a minute. With the help of Didier Ronier, we did some modeling, and that's actually how doxo-EMCH was designed. We looked at the cleft of this cysteine 34 position. And this cleft is about 10 to 12 angstrom deep. And that's why we chose this, the length of this spacer with 5 carbon atoms. And you can see in this modeling structure, there are actually hydrogen bonds which stabilize doxorubicin, which sits very close to the protein surface. Binding takes place very rapidly within 5 minutes to cysteine 34, but at pH 5, you have a very short half-life. Doxorubicin, the original doxorubicin, is released within 25 minutes at 37 degrees. So the mode of action basically looks like this. You have doxAMC, which is now reacted, circulating. It reaches the tumor cell and is then taken up by adsorptive or unspecific endocytosis. You have an option, because we know that also in the extracellular environment, pH can be around 6 to 6.8, that there is some release of the drug extracellularly, but primarily the uptake will be by endocytosis. The pH in endosomes, lysosomes goes down to 4 or 5, and then doxorubicin is released in the cell. This is what a typical animal experiment looks like, with a straightforward comparison of the MTDs of doxorubicin versus doxa-EMCH. This is 3 times 24 milligrams per kilogram with respect to doxorubicin equivalents. For doxorubicin, it's 2 times 8. This is a mouse model. The control, as you can see, the tumor volume is increasing by a factor of 5. Doxorubicin, some effect, but the tumor volume still increased by a factor of 3. But at this dose, which is the MTD, you see complete remissions basically in all animals. So that was now, after we had repeated it in some other models and saw basically the same result, was the initiative to move it into the clinic. And this was all done at the Tumor Biology Center and gave me a good chance to get some experience in project management to coordinate all these steps. So we had done all this. The next step was to look how could we scale this up. Most of it was done in my lab, but then when we had to do it under GMP, we found partners. The 100 grams at this time were done by Nicomet. The in vivo studies we all did ourselves. This also in collaboration with the university. The GLP toxicity we did with Aventis Protox, which was close by in Frankfurt, in 3 species, mouse, rats, and dogs. The shift here in the MTD in these animals was, compared to doxorubicin, it was a comparison to doxorubicin in all of these species, was 2.5 to 5-fold. The dog being the least sensitive species, then the mouse with 4.5, and the rat that is the most sensitive organism for doxorubicin, it was 2.5. And then we did the phase 1 study, and this was good enough to license it out first to an American company based in New York, Innovive, which was later bought by Citrix, who are now doing the clinical development. This is the result of the phase 1 study at the Tumor Biology Center. It was a classical dose escalating study. The normal dose you can give for doxorubicin is between 40 and 75 milligrams per square meter. These are the side effects you will have. And of course, the aim of a phase 1, first of all, is to look at side effects. Second endpoint is anti-tumor activity. A lot of acute toxicity, but also fatigue, alopecia, and cardiotoxicity. We started at 20 milligrams per square meter, dosed up in steps, and at 200 we didn't see any side effects. So this is already a great shift. The first side effects appeared at 260. These were basically neutropenia. At 340 milligrams per square meter, mucositis also turned up, and this was then the dose-limiting toxicity. So the MTD was defined as 260, which is basically, compared to 75, a factor of 3.5 to 4. So this is important because the shift you see in animals, for instance in mice, is comparable to what we had seen here in humans. That was one of the goals of that study. Regarding tumor response, this is what we could evaluate in 30 of the evaluable patients. 12 patients had progressive disease, basically at the low doses. 13 patients had stable disease. 2 minor remissions. That at that time was defined as 25% reduction of all tumor burden, and 3 partial remissions, over 50% tumor reduction, and these were achieved in small cell lung cancer, mammocarcinoma, and liposarcoma. One result is shown here for the breast cancer patient. She had no longer responded to conventional therapy. She had a large liver metastasis here. And after 4 cycles only, you can see there is a clear partial remission over 50%, and that progression was stable over 6 months. We then, as I mentioned, licensed it out. The next steps you do are scale-up, optimization of galenic formulation. You want a commercially viable formulation at that time. You want to get for phase 2 studies your documentation in place with the FDA and also with the European agencies. And this is basically how it is used worldwide now. It's made now by Baxter Oncology in these vials, reconstituted with Ringer solution, and that is what is infused to the patients. Citrix decided to do a preliminary study on-site in Los Angeles in 18 patients with primarily sarcoma. They moved on then to soft tissue sarcoma at the dose we had found, MTD of 260 milligrams per square meter. And there are further ones starting still this year. I'll talk about that in a minute. This is a result of that small study. These are all sarcoma patients. And you can see this is a waterfall plot showing the response. Now, you can see these patients at 260 did not respond. This is more or less stable disease. But these above minus 30%, that is now the new definition under RECIST, counts as a partial remission. And what is noticeable, there were 5 patients who actually responded who had already received an anthracycline, either doxorubicin, epirubicin, or Doxil. So that was an interesting thing to see, because normally you wouldn't expect that. This is now the beginning of the phase 2 study, just to give you some information on soft tissue sarcoma. The soft tissue sarcoma is an orphan drug disease. It affects muscle, fat, blood vessels, nerves, tendons. To give you an idea of the number of cases per year in Europe and the US, it's 40,000 and 13,000 cases in the US and Europe. And doxorubicin is actually the only approved drug for treating soft tissue sarcoma. But all patients under doxorubicin ultimately progress, even if after that you try other therapy options. This is how the phase 2b study was set up, which is all patients have now been enrolled. Of the 120, 80 patients were enrolled with doxorubicin at the MTD of 260. Arm 2 was at the curative dose, the highest dose you can go for with doxorubicin, 75 milligrams per square meter. It was done in 13, and is still ongoing, clinical sites in the US, Hungary, Romania, Russia, Ukraine, India, and Australia. So that's a large network you have to set up for these not so common tumors. And the endpoints were tumor response, progression-free survival, and overall survival. Cycle every 3 weeks, up to 6 to 8 cycles. This is an interim report. More will come out at the end of the year. The dramatic difference is there are 22% partial remissions for the aldodoxorubicin-treated patients, and no response for any patient receiving that dose of doxorubicin. The stable diseases more or less are equal. Now you have to consider, when you talk about a partial response, It's defined now as minus 30% of reduction of all tumor lesions in that patient, but it can of course also be minus 80%. That still counts as a partial remission. What you see here is one of the patients of that study. This is one of the sarcoma lesions, and you can see after therapy, after 8 cycles, it has disappeared. But the shrinkage of all target lesions was indeed much higher. It was 80%. So at the end of the year, we will have data on the progression-free survival, and then during 2014, overall survival will take longer, maybe in spring. We will have data on that. The phase 3 has already been granted by the US, so that is scheduled for the beginning of 2014. And Citrix are now extending it this year into 2 other indications, into glioblastoma and into Kaposi sarcoma. And I want to talk about, to the end, as the last indication, about brain tumors. Glioblastoma multiforme, it's the most frequent brain tumor among tumors of the brain. Affects 12,500 in the US annually, to give you an idea of how common this cancer is. The median survival with all combinations of therapy, surgery, radiotherapy, chemotherapy is 14 months. 5-year survival is poor. What we did, we compared doxorubicin and aldoxorubicin in an intracranial xenograft mouse model in a typical cell line, the U87 glioma cell line. And this was luciferase tagged, so we could follow the growth by luminescence. And this is what you see. This is after 8 days before therapy for 8 animals per group. This is the control, this is doxorubicin, this is aldoxorubicin. Therapy started on day 12. So after 19 days, this is after a week of therapy. You can see already animals are dying in the doxorubicin, in the control, and the tumors are growing. They're quite large. Also, the doxorubicin group is not responding well. 2 animals have died, but the aldoxorubicin, there's a much better response, as you can see here. After 28 days, basically 80% of the controls have died. For doxorubicin, 90%, but the aldoxorubicin-treated animals are still all alive. Compared to these large tumors, there is still a tumor shrinkage. The Kaplan-Meier shows you the survival of that experiment. We started treatment on day 12. You can see these are the doses we used, MTD of doxorubicin 2 times 8 milligrams per kilogram, and basically doxorubicin is inactive in this model. Aldoxorubicin, however, at the MTD is significantly more active. Only 3 of the 8 animals have died on day 60. Now, we were very surprised by this result because, of course, there's a blood-brain barrier, which is hard to cross. It's intact. It's not leaky. And there's no evidence that tumors are leaky. But when we looked at the tumors, we were surprised to see that aldoxorubicin was transporting doxorubicin to the brain tumors. This is a separate experiment. We injected 24 milligrams per kilogram into a tumor-bearing mouse. And you can see there is, because doxorubicin is fluorescent, when you then irradiate this, there is no fluorescence in the tumor. But there is definitely fluorescence in the brain tumor, but not in the healthy brain. If you then look histologically at these parts, this is what you see. This was histologically marked with different compounds, DABI for cell nucleus, CD31 as a marker for vessels, for angiogenesis. This is for doxorubicin, and this is vimentin. This is a filament marker showing you the structure. For the aldoxorubicin tumor, you can clearly see the normal brain tissue. There's no uptake of aldoxorubicin, and there's not any really signs of angiogenesis. This brain tumor, however, the glioblastoma, there is angiogenesis, and you can see by the overlapping of these two colors, doxorubicin is going to the cell nucleus of the brain cells. Here's the border for doxorubicin. You can see it, but you don't see any sign of doxorubicin entering either the tumor or the normal, healthy brain. And that's now the incentive for starting a phase 2 study in glioblastoma in the US in these 3 sites with 30 patients, and that will start in 1 or 2 weeks in December. Now, I'm going to finish with some thoughts on the mechanism of uptake of albumin in solid tumors. What is perhaps special about albumin? This is an experiment taking you back in time to 1986, when a young Japanese scientist injects this dye, Evans blue, into a tumor-bearing mouse. And after a very short time, these tumors growing subcutaneously turn blue. And you all know this man. It's Hiroshi Maeda, paper published in 1986. The reason is not that the dye has an affinity for the tumor. It binds rapidly to albumin, and albumin transports this blue dye to the tumor. And this is what you're seeing regarding the pharmacokinetics of the Evans albumin complex. And this is the uptake now in the tumor over time. There's a maximum around 24 to 48 hours, and as a comparison, very little uptake in muscles. Now, this is investigations which I did in my own lab a couple of years ago. It's a very reproducible experiment. You're injecting exactly the same amount as Hiroshi did, 10 milligrams per kilogram, and then you can draw out the blood, you can centrifuge it, and you will see after 1 hour you get exactly his value, 85 micromolar of this dye is circulating. Strangely enough, this publication, Cancer Research, good impact factor, was ignored for at least a decade. It was picked up maybe around here, because of course we don't know why. He was using albumin, and nobody was interested in albumin. Of course, this was still the era of liposomes, of antibodies, and the emerging era of synthetic polymers. I think you're all acquainted with the EPR effect. I'm just showing you it as a short memory effect. You basically, in healthy tissue, in most of healthy tissue, have an intact endothelial layer, shown here. So small molecules can enter, but large molecules can't. And tumor vessels in certain parts are leaky. They have these pores which can be very large, 100 to 500 nanometers, so that albumin but also nanoparticles can enter. And then you have a second effect. In comparison to healthy tissue, the tumor tissue in general does not have a lymphatic drainage system, so these particles are not transported away, and you have a retention effect, and that's why this effect is called permeation and retention effect. And it is a physiological property of solid tumors. This is when you do it yourself. This is in my lab in a pancreatic model. After 6 hours of injection, this is what you see with Evans blue. when you dissect the mouse, you see everything is blue because it's circulating in the bloodstream, so you have to infuse it with saline to get all the evidence blue out of the bloodstream, and then you can actually look at the tumors and the organs. And you can see the tumors here now are stained blue, and with the liver and kidney you can't really see it. If you extract the Evans blue again, basically what Maeda also did, this is what you see. You see that you can extract it, the tumors become yellow again, and there's a peak around 24 to 48 hours in that color. Now, one of the problems actually of drug delivery is not necessarily only getting as much as possible of the drug into the tumor. It is also about getting it everywhere into the tumor. Because tumors are incredibly heterogeneous. They have necrotic areas, they have a large interstitial fluid pressure, they build up hypoxia, and the vascularity and permeability varies within the tumor. The EPR effect probably is universal for solid tumors, but it's not universal in all parts of the tumors. There will be areas where there's no EPR effect at all. We can assume that. One of the things which was already known when I entered into the albumin field, I had actually no idea even of the Maeda experiment, but ignore that. I had no idea of this work from Schnitzer. This receptor, the GP60 receptor, had already been found, and this is a receptor called albondin, and it binds albumin on the surface of an endothelial cell, and then you get transcytosis, and that's how albumin enters the tumor interstitium. This is one other method besides the EPR effect. And then you have a second protein called SPARC. This is secreted protein enriched in cysteine. It also binds albumin with high affinity, and this, again, comparable to the EPR effect, is now retaining albumin in the tumor interstitium before it reaches the tumor cell. This is also interesting because the pH goes down in these vesicles, so if you imagine DOX3-EMCH, there is also the chance that doxorubicin is released in the endothelial cell, and then you also have an anti-angiogenic effect of this drug, not only an effect on the tumor cells when it reaches the tumor cells. SPARC is one of the markers you can use preclinically and clinically. And this is a collaboration with Oncoplex, who have a nice method of determining SPARC, and this is shown now here for breast cancer, stroma, lung cancer, and our own samples from the experiment I showed you, the pancreatic cancer. And you see the levels here are between 100 and 900 attomole per microgram of tissue, and you see the pancreatic SPARC levels are around here. This is important because SPARC can now be used and has been used clinically to actually move drug delivery into personalized medicine. This is a phase 1-2 trial with the combination of gemcitabine and Abraxane. And what you're seeing here, you're seeing all of the patients regarding their survival selected for low SPARC expression, and that survival in that population was only 8 months. On average, you will have with gemcitabine 10 to 12 months. But those patients who were expressing high SPARC levels had 18 months. That's 6 months more than the average you would get with gemcitabine. So that's a very good marker to use albumin in future for any kind of clinical trial. And to finish off, and to show you the importance of GP60 and SPARC, this is seminal work 2007 from Dennis et al., who did a very interesting experiment. He looked at tumor distribution of an IgG. This is trastuzumab, Herceptin, which you well know, addressing breast cancer. And he used a single-chain body, which was also interacting with the HER2 Neu receptor. And he designed a single-chain antibody, this one, and put an albumin-binding peptide on it. And this would result in this one, 90 kilodalton albumin with the single-chain body attached. And then he compared the uptake. And what you see here is the uptake. He labeled it with FITS. And this is what you see. The IgG and the area under the curve over time is here, and it's very comparable to this one. We know single-chain antibodies, because they are eliminated much faster, they don't reach the same area under the curve in tumors. But these are identical. The big difference, however, being, if you now look at the distribution within the tumor, you can see that the IgG, and this is the commercial product, is unevenly distributed within the tumor. You can see that also down here in the bars. But the albumin product with the same fragment here as in Herceptin shows a much more even distribution. And that becomes important when you talk about what kind of effect you want to reach. This is the last slide. I've concentrated on oncology and drug targeting, talking about Abraxane and the drug we developed in my group, and now in collaboration with Cytrex. There are also many other uses which have reached the market and also doing very well. I'm just going to mention diabetes. There are two products now from Levemir, which are, again, very simple technologies. They bind, for instance, Levemir binds a fatty acid to insulin. That's all it is. And that is injected into the bloodstream. The fatty acid binds to albumin, and then you have a long circulation time. And that allows just one administration per day for a diabetes patient, which is a great advantage, especially for children, because they have problems in controlling their sugar levels. And with that, I'll finish. But because I started in Italy, I'm going to tell you what is the most important thing I learned in those 2 years in Florence. Italian institutes do not have coffee machines. I don't know if it's different here in Pavia, because there are the bars. Everywhere is the bars. So typically, on a typical day, the Italian colleagues would go down to the bar, which was actually right 50 meters away from the institute. And we would have a cappuccino or an espresso, and they would always pay. They would always pay. And I said, I'll pay the next time. And they always laughed at me. And then they said, Felix, tu sei così tedesco. So take this as a compliment. Thank you.